Good evening, everybody. A warm welcome to this evening's Zoom webinar on behalf of St Hugh's College. My name is Robert Verlaine. I'm the Senior Tutor of St Hugh's. I've been Senior Tutor since September last year, so I'm new to the college. But I've been made to feel extremely welcome by everybody I've encountered, students and staff alike. It's a very great pleasure this evening to be able to introduce our lecturer, Dr. Chris Poole, for his paper on preventing epidemics. What do ants have to teach us? You will see from my camera that I have my daughter behind me over my shoulder. She's also keen to learn about ants. She may not stay the course, but she's under strict instructions to be very quiet and listen attentively. Dame Ailish Angelini, the principal of the college, would normally introduce these lectures. As many of you may know, she's currently on secondment to lead a public inquiry. She sends her warmest greetings to everybody, and would be may very well make it as a guest listening in. It's wonderful to see so many members of the St Hugh's community here from right across the world, and I'm grateful that you've been able to join us for what will be a very thought-provoking session. You're very welcome. You will have had a chance to find out a little about Chris Poole from the fascinating article that he's published in the most recent edition of the St Hugh's magazine, The Chronicle, and I'm looking forward to finding out even more from this evening's talk than he was able to publish there. We're very fortunate in St Hugh's to count Chris amongst our stipendiary lecturers. He's a lecturer in animal behaviour at the university's Department of Zoology, and his research focuses on how social animals overcome the problem of disease. Chris holds a master's degree from my old stamping ground, from Royal Holloway. I spent 18 years of my career there before going to Bristol and then joining St Hugh's. So it's very pleasurable indeed to welcome a colleague from there. He started studying ants in 2011. He went on to investigate how ants detect and then prevent infections from turning into colony-wide epidemics during his PhD at the Institute of Science and Technology in Austria, before returning to the UK to undertake postdoctoral work again at Royal Holloway, where he focused on the memory of foraging bumblebees. There will be an opportunity to ask questions of Chris. During the webinar, please take the opportunity to put questions by typing into Q&A. We'll do our very best to get to as many of those as possible in the time available to us. I'll hand over to Chris in just a second, but before I do so, I really do want to express my gratitude to everybody who's made a donation to the St Hugh's Discretionary Fund, which helps us to support areas of greatest need in college. When you booked your ticket for this event, we are very grateful indeed for your generosity, and we are grateful to those of you who return time and time again to these lectures and are generous every time. This is very much appreciated. I will hand straight over to Dr. Chris Poole.
Yeah, thank you very much, Robert. I'm just making sure I'm sharing my screen. Okay, that should be sharing now. So yeah, thank you very much for coming tonight and for coming to see my talk. I'm really excited to talk to you about the work which I've done sort of thus far in my career and what I hope to achieve at Oxford. So I joined Oxford relatively recently. So I'm really excited to begin sort of my full-fledged independent research career here. So today I'll be talking about how ant colonies are able to cope with epidemics and potentially what we might be able to learn from them as well. So I don't really need to labor this point too strongly given the situation that we've been in the last 2 years. However, looking at a picture like this very crowded train platform, it's easy to imagine how infectious diseases such as COVID-19 can spread, fairly easily and rapidly from infectious to susceptible individuals. And that's because infectious diseases are able to take advantage of the sort of social network that exists between individuals and so spread from people who are currently infectious and infect susceptible individuals. And it's that close proximity and social interaction between people that facilitates that transmission. Now, humans aren't the only sort of society that has to deal with infectious diseases. Many social animals suffer from epidemics and infectious diseases. And I study this primarily in ants and other social insects. So social insects, just like humans, live in these very densely crowded societies, and they actually also share things like they share liquid food via mouth-to-mouth interactions, and so in principle they should be really susceptible to disease transmission. So that's what I'm going to talk a bit about now. So why are social insects susceptible to disease? So as we sort of just maybe just to start, then we should actually just clarify what we mean by social insects. So specifically here, I'm talking about the social wasps, the social bees. So that's the honeybees and the bumblebees, the ants, which are all social, and then the social termites as well. So this is what I mean when I talk about the social insects. And so like we just said, social insect societies, just like humans, have the issue that diseases can spread through the kind of social network of the social insect colony, and potentially cause epidemics. Now, the situation is exacerbated in social insects because of numerous factors. And a principal factor is that all the individuals within the colony are actually related to one another. So in case you're not aware, social insect colonies are typically founded by a single individual. So that's usually what we call the queen. So you can see a queen ant here. And she generally, every other individual in the colony is typically her offspring. So all of the workers are the sort of daughter offspring of this single queen. And what that means is that they all share essentially the same genes, the same genotype. And so a pathogen that's able to overcome one individual's immune system is likely to be able to overcome all the others in the colony as well, because they have the same genotype. And so there's a risk there that if one of them gets sick, they could all potentially get sick. So this is one of these factors that makes them potentially very susceptible to infections. Another factor is that there's overlap in generations within colonies. So the current sort of nurse cohort of workers takes care of the next generation of workers. These are the brood here, and they'll become the next sort of worker generation. And there's a risk there of disease transmission from the current worker cohort to the next cohort. And so you can imagine how diseases then could potentially persist in colonies over time. So some of these colonies are very long-lived. I think the record is about 50 years or so in termites. So there's the potential there that once an infection gets into the colony, it could be very hard to remove because it can actually transmit across generations within the colony. Additionally, social insects often work very hard to maintain sort of stable nest conditions. So particularly things like honeybees and termites put a lot of they design the nest in such a way and to promote gas exchange and to maintain a sort of homeostatic temperature. And this is ideal, obviously, for raising brood and for the insects themselves, but it kind of acts like an incubator as well. So it's ideal for microbial growth. So it might promote the growth of pathogens as well. So in principle, then, social insects should be, really susceptible to disease, and we would expect perhaps to see epidemics in social insect colonies all the time. But in fact, disease outbreaks are seemingly rare in social insect colonies. And, this might simply be an artifact of, a sampling bias. So maybe we just don't have good data on how many social insect colonies actually die out due to diseases. But there is, fairly good evidence from founding colonies. So where they've just been established by a founding queen, a lot of those colonies actually die out due to infections. Some other sort of important lines of evidence come from the fact that for many decades now, people have tried to control, or the hope is that we might be able to control pest social insect species. So particularly things like termites and leafcutter ants, which cause huge amounts of sort of economic damage to crops and people's houses. There was great hope that we might be able to use microbial pathogens as a biological control to eradicate such pest species, because they should in theory be able to spread within the colony and sort of continually infect individuals and hopefully wipe out the entire colony and the damage and stop the damage which these insects are causing. But in sort of 50 years of trying to come up with a biological control that can be applied in a sort of industrial setting, it's failed again and again as far as I'm aware. So it's really interesting. It really suggests that these social insects seem to be somehow immune or have sort of solved the issue of disease. So the question, which we started with, why are social insects susceptible, really has become, why aren't social insects susceptible to disease? And that's what I'm going to explore for the next part of the talk. So one of the reasons is perhaps that they've just been, unlike human societies, social insect societies have been co-evolving or coexisting with their pathogens for millions of years. So this is a really nice picture of an ant trapped in amber, and this unusual structure here is actually a parasitic fungus growing out of the ant. And so for at least 50 million years, we know that they've been being infected by parasites and pathogens. And so perhaps they've just had a really long time to evolve adaptations to counter the effect of pathogens and to solve this problem. And the way that they solve this is sort of through the same way that they are able to achieve these amazing feats of architecture. So this is a termite nest, which kind of looks like a cathedral. How they're able to collectively forage and find food, how honeybees are able to produce kilograms of honey every year, and they're able to also make things like rafts to actually traverse water. And that's through the principle of kind of collective and self-organized behavior. So what we're seeing in these social insect colonies is that they're able to collectively fight disease together through this kind of self-organized emergent behavior. So I'm going to give some examples now of collective disease defensive social insects. And to start, I want to introduce this idea of social immunity. So within the colony is obviously comprised of individuals, and each individual has its own like personal immune system, so it's able to mount an immune response to an infection. But it can also perform behaviors that reduce its risk of infection. So it could groom itself, for example, or avoid areas of contamination. And we call this kind of its individual level immunity. And obviously the colony is comprised of many individuals. And so there's this kind of additive effect of these individual immune defenses. What we see in addition to this individual level is kind of a social immunity, this kind of secondary layer of protection in a way that results from the collective behavior of individuals within the nest. And we call this social immunity, and it's often referred to as colony level disease protection. And it emerges through the collective self-organized behaviors of the individuals. It can be prophylactic, so it can be something like nest structure, which is always in place and always active to try and reduce disease transmission or uptake, or it can be induced. So for example, this ant here, which has been painted red, has been exposed to some fungal spores, and you can see its nestmates are grooming it. So this behavior is elicited by the presence of a pathogen, so we call it induced. And so these behaviors are primarily behavioral, but they also have sort of physiological or what we call organizational components. So I'm going to talk specifically about some of these, some of the ways in which social insects are able to prevent epidemics in their societies. And I'm primarily going to focus on ants and the fungal model system, which has been used extensively to study collective disease defense in social insects. So the pathogen which we typically use is this generalist fungal pathogen called Metarhizium brunneum. So you can see here this green kind of growth on the petri dish. This is the fungal spores. And the model ant system, which we use usually, there's two species of garden ant, which we use. One of them actually is invasive in Europe. So this is the life cycle of the fungus, of the parasite. So spores are typically picked up from the environment by individuals exploring that environment. So that could be whilst they're foraging or whilst they're expanding their nest. So these spores are actually found in the soil. And I believe I came across a statistic that 1 gram of soil contains roughly 6,000 of these fungal spores. So they're really prevalent. And the ants are presumably coming into contact with them quite often. So once the spores get onto the cuticle, onto the body of the ant, it takes a few days for them to actually penetrate into the body. First of all, they loosely attach to the cuticle of the ant, and they produce this kind of germ tube, which you can see here. And that eventually, using enzymes, it makes a hole in the cuticle and it causes an internal infection. And if that infection can overcome the immune system, the pathogen will actually kill its host. It has to kill its host, in fact, to then produce new spores, which can continue the cycle. So once the host dies, the fungus basically breaks out of the ant's body and produces these packets of spores, which you can kind of see illustrated here. And each of these spores contains thousands and thousands, each of these packets, sorry, contains thousands and thousands of spores. And so you can imagine how, if the ants don't do anything, and they allow these infected individuals to die and to become infectious in the nest, how this could very quickly lead to a pandemic or epidemic, sorry, within the colony. So we're going to look at the ways in which ants can basically disrupt this cycle. So sort of one way in which they can do this is to be very proactive about their waste that they produce. And you can imagine a colony that contains millions of individuals will have quite a high turnover rate in the number of workers that are dying. And so we actually see individuals specialized on undertaking, we call them undertaking ants. And what they do is actually carry their dead, usually outside of the nest, to sort of specialized graveyard areas. But some species also have specific chambers within the nest, which act as a graveyard. And here, for example, you can see a queen ant here. She founded a colony with another queen ant, which had an infection. And it's just a really nice illustration of, essentially, she's kind of chopped up the body and buried it here using sort of plaster and the other nest material which she has, and really segregated that potentially infectious individual from her brood. And these graveyards can actually be rather large. So this is an example of a graveyard which we came across in Panama. And you can see, it's sort of a meter or two across, actually. So a huge amount of waste being produced. And it makes sense to segregate your waste and your dead, because then you can limit and control how much you interact with them. So if ants were to just dump their dead individuals sort of anywhere in the environment, there's a real risk then that they re-encounter those corpses and potentially pick up infections from them. Whereas if you concentrate them and have just a few specific undertaker workers that interact with those corpses, it reduces the risk of reinfections into the colony. So that's one way in which they can sort of prevent or break this pathogen's life cycle. Another is to limit how they interact with one another. So we might naively think that within a colony, every individual at some point comes into contact with another individual, and that they're all, through one way or another, they're all connected through what we call a social network. And for a long time, it's been hypothesized that actually they modulate their interactions or their social network in such a way that it limits or reduces the risk of disease transmission. Now, a few years ago, a group in Switzerland, Laurent Keller's group, kind of showed using a novel sort of tracking technique that this doesn't, there is evidence to support this. So what they did was to put these little QR codes onto the back of every ant within the colony, and to then track over several days who interacts with who and sort of where they spatially distribute themselves. That's what you can see an example of here in the background. So you can see the brood here, and I think queen is here somewhere. And when they analyze this data, they produce a social network that looks like this. So every dot is an ant, and the line is the lines are the connections between those ants. And sort of what in support of sort of a modulated interaction net hypothesis is what we see is the queen here in the center, this dark blue dot, as she's surrounded by a retinue of nurses, and they mostly interact with one another. And when they interact with other individuals within the nest, it's primarily with this kind of second group of workers called the cleaners, and they're ones performing sort of in-nest tasks. And then the third group, which is the most isolated in the social network and the most tightly clustered, are the foragers. And the foragers are obviously the ones which are leaving the nest and the ones which are most likely to come into contact with diseases. And so it makes sense that they should be the most sort of isolated and separate from the rest of the colony. And in particular, you want to make sure that they have few links, or the colony wants to make sure that they have few links with the queen and the nurses, because they're taking care of, obviously, if the queen dies, the colony dies, and the nurses are also taking care of the brood as well. And you don't want to get them infected. So contrary to, everybody interacting with everyone, we do see this kind of modulated social interaction network. And it's been sort of shown through simulations and models that this sort of standing baseline level of interaction is actually, would actually prevent diseases from spreading, or at least limit their spread. But a few years ago now, Natalie Stromiek from the University of Bristol actually performed some experiments using this tracking system, where she actually introduced a pathogen and actually observed how this network changes. So this is these pre-exposure networks, so before there's any pathogens introduced to the system. But in gray here, these gray dots are the ants which are going to be exposed to the disease. And what you see after exposure is that these pathogen exposed individuals, so these now have these fungal spores in their body, we actually see self-isolation occurring. So they tend to isolate themselves from others, and so restrict their interactions with the rest of the social network. And what's interesting is that there's a group of caregivers, and we'll come back to caregivers in a moment, that they tend to interact with most. And then we see proactive distancing of the rest of the colony, so where they limit their interactions with these individuals, but interact more with one another. And the coloration of these dots here indicates how likely it is they are to be exposed to the pathogen. And you can see that in this cluster here, they're all kind of this more dark sort of purple color. So that's least likely to be exposed to the pathogen. So here we see an example of prophylactic social immunity, in that the colony social network is designed or in such a way as to limit disease spread, but also induced. So actually, when there is an infection present in the colony, they change their social network to try and reduce the risk of disease transmission within the colony. So we've seen how good nest hygiene and organization, that's what we call organizational immunity, by the way, this kind of modulated social network. So we've seen how nest hygiene and organizational immunity can limit the risk of infections within the nest. But if an individual is exposed, so as we saw previously here, these individuals are exposed to the pathogen, and we see there are caregivers. So what are these caregivers doing? What can the colony do to prevent those exposed individuals from actually contracting an infection? And if you recall, I said at the beginning that these spores loosely attach to the cuticle to begin with. And what that means is that ants can actually, these caregiving ants can actually perform what we call sanitary care, where they actually groom and remove these spores before they can cause infections. And what's interesting is that this grooming has kind of a dual function. So on one hand, they're mechanistically, physically removing the spores so they can't prevent infection. But at the same time, the ants are chemically disinfecting those spores as well. And the way that they do that is through producing a venom. So the species we work with, primarily Lasius neglectus, has what we call an acidopore here at the tip of its abdomen. So it's a little gland opening here. And this is connected to a venom gland, which contains primarily formic acid. So about 60% or so of this poison gland content is formic acid. And then some of it is also acetic acid. And then there's a bunch of other compounds in there as well. And we're not 100% sure what they do, but we think that they help the formic acid to stick to surfaces. And this gland content has a really low, with all these acids in there, unsurprisingly, it has a really low pH of about 2. So really, really acidic, really acidic. And what's interesting is that they use this venom as a chemical disinfectant. So this is a picture of a nest, ant nest, and what we've done, or what we keep the ants in the lab anyway, they treat it as a nest. So these are kind of mimicking the chambers that you would see in the wild. And we line this nest with pH sensitive paper, so it turns pink when the ants are spraying their acid. And what you can see is 24 hours after the ants have moved into this new chamber, we actually see patches of pink here where they've been spraying their venom. And so it really seems like they actually use this venom as a sort of prophylactic chemical disinfectant to disinfect their nests. But as I was saying, they also use it during grooming as well. So this is a nice video from Simon Tragaust, who was also at IST Austria as well. And what he showed is that instead of actually spraying this venom directly onto pathogen exposed brood, what the ants do is actually suck it up into their mouths and then reapply it whilst they're grooming. So here you can see the ant has sort of bent its abdomen under and it's sucking that venom out of the acetopore and then spitting it out basically whilst it's grooming. And we think the reason why they do this is that the venom, is really acidic. It can also kill the ants. And so by taking it up into their mouth and sort of carefully applying it where it's needed, they reduce the risk of any sort of collateral damage or self-harm when they're using it. And we actually showed in a follow-up experiment that indeed, if they're not cautious about using it, they can actually cause harm to the brood that they're trying to protect. So it does seem that by being sort of judicious about how they use the venom, then it makes sense for them to sort of carefully apply this venom whilst they're grooming. And interestingly, the spores which they're removing, they're actually storing they've got essentially like a sieve in their mouth, which prevents them from swallowing the spores, and they collect in in a ball inside their mouths, and they actually mix this with the poison, with the venom which is in their mouth. And there's some really nice pictures of them sort of spitting this ball out, and then when you put that ball of spores onto pH sensitive paper, you see it become sort of pinker and pinker. So it's actually got that acid sort of wrapped up inside these spores. And if you try and play out those spores and grow the fungus, it won't grow anymore. So they are, as I sort of said, they're mechanistically removing the spores and chemically disinfecting them at the same time. So so far we've focused on nest hygiene, organizational immunity, and how ants are able to prevent infections. And I just want to sort of pause and focus on these caregivers again. So obviously there's a risk here that, like people who were on the front line during the ongoing COVID pandemic, individuals on the front line obviously were contracting COVID as well. There's a real risk here as well in the ants that the ants, whilst they're performing this care, actually contract infections themselves. And what's really interesting, so another one of my colleagues, Matthias Conrad, performed a really nice study showing that essentially what happens is that these caregivers contract an infection, but then they become immunized from it. So here they use fluorescently labeled spores. So that means we can actually identify them under the microscope because they fluoresce when you shine a laser on them, essentially. And so that's what you can see in red here. And these are images from the directly exposed individuals. So you can see these spores collecting here on the joints and on the antennae and on the head here. And what's interesting is that you can see like one spore here. This is on the caregivers now, on the nestmates. You can see one spore here on its antenna, another one on the leg. And so they are actually contracting some of these spores whilst they're performing this grooming behavior. But the dose they get is very, very low. And so what they did was to actually sort of crush these ants, these directly exposed ants, and plate them out. And what you see is that in almost all cases, they actually have active spores within or on their body. And the same is true of the nestmates as well. So these little spores which they contract seem to actually be causing like micro infections within the body. And you can see the majority of them actually, that's what you can see on the plates growing here, the spores. The majority of them actually contract these infections, but they're not lethal. So about 98% of these caregivers survive providing care. Only about 2% actually contracted infections. And what's really interesting is that when you then expose these ants to a lethal dose, they actually then survive. So this graph shows this is individuals exposed where they were treated with a sham control or UV radiated spores, which can't cause infections, or they were these caregivers. And then all three of these groups were exposed to a lethal dose of the pathogen here. What you can see is that those individuals that had this kind of immunization from having these micro-infections, they actually survive longer and have a higher survival in the end than these other individuals. So essentially they are immunized against the disease. Okay, so finally then, what happens at this step here? So if an individual contracts an infection that's successful, so it's overcome the immune system and death is essentially imminent, what can the colony do to prevent those infected individuals from producing new spores, billions of new spores within the nest that can then infect the rest of the colony? And so that's what I'm going to talk about next. So how do ants prevent successful infections from becoming epidemic within the colony? So to study this, we decided to focus on the brood. And the reason for that was that we didn't want to, we just wanted to study the response of the colony rather than, having also, like if we expose an individual ant, it will perform some behaviors itself, which will, interfere a bit with what we want to observe, which is the colony's response. So if we focus on the brood, they can't really perform any behaviors. So we decided to look at brood infections, and we specifically focused on the pupa. So the pupa is the stage which comes, so just in case you're not aware, ants, first of all, they lay eggs, or the queen lays eggs, and these eggs hatch into larvae, which go through multiple molts, and they get larger and larger. And eventually the larva spins a cocoon around itself like a caterpillar, and it undergoes metamorphosis as a pupa, which is what you can see inside the cocoon. And eventually that hatches, obviously, as an adult ant. And so we focus on the pupal stage here. And the reason for this is that out of all the brood, the pupa is the only one which is actively removed from the nest. And the reason for this, so this is a bit of a shaky video that I took from a house that I used to live in. And what you can see here, the ants are actually carrying pupa out of their nest, bringing them into the house and then back into the wall of the building. And that's because the sun was on this wall and they wanted to incubate their pupa and actually speed up metamorphosis. And you can imagine how during this passage, the pupa are leaving the safety of the inner sanctum of the nest, going out into the external environment and potentially being exposed to pathogens. So it makes ecological sense to also study the pupal stage and how ants prevent infections there, or how they deal with infections there. So the kind of experiments that we do are all lab-based. So we typically have a few ants in a petri dish, and we kind of mimic the kind of settings that they normally live in. So they have a little nest area, some water, and we keep them on this plaster substrate so that they can't dig into the plaster, but it remains moist like the soil. And we cover up the little nest area with some red foil, because the ants can't see red light, so to them it's dark, but it allows us to still see in. And this is sort of what it looks like inside. So we have these ants, and they are usually given a pupa when we perform these experiments. And so the first experiment which we performed was to see how do the ants respond when the pupa are exposed to pathogenic fungal spores. So this is the same pathogen which we've just been discussing. You can mix the spores in a solution and apply that to the cocoon. That's what you can see here. And what's really interesting is that after a few days, we observed this completely novel behavior which hadn't been observed before, which we called unpacking. And that's where the ants basically cut the pupa out of these silk cocoons, and they expose the nude pupa inside. And what's interesting is that, so this graph shows the proportion of pupa being unpacked from their cocoons over time. So on the x-axis here is day. And we used 3 different dosages of the pathogen, so a really high dose, a medium dose, and a low dose, and we had a control as well. And in the control group, as you would expect, well, as you might expect, we don't see any cocoon removal, no unpacking occurring here. So that suggests that this is a response to the pathogen. And it's also dose dependent. So we see more of this behavior when more spores are applied. And this kind of suggested to us then, perhaps this is a response to infection. And that's also because it occurs at around day 2 at the high dose, and a bit later in the lower dosages, but it doesn't occur immediately. So it doesn't occur simply due to the presence of the spores, but it occurs a few days later. And we know that it takes a few days for this pathogen to actually cause an infection. So we speculated that the ants are performing this behavior in response to an infection. And interestingly, when we take these nude pupa away from the ants and we incubate them for fungal growth in the absence of ants, we find that about 85% actually sporulate and produce new spores. So indeed, they actually had an active internal infection, and if left without any further processing by the ants, they will become infectious. So it seems that this kind of unpacking, this cocoon removal response is indeed a response to infection. So we wanted to understand why the ants perform this behavior. So we filmed the ants over the course of a few days. So we gave them, again, these pathogen exposed pupa, and we recorded their response sort of before and after they removed the cocoon. And again, we do this on pH sensitive paper so that we can observe any acid spraying from the acetopore that goes on. So I'll play this video now. And what you see, so here the ants are removing the pupa from its cocoon. And what they start to do is actually spray the pupa directly with its venom, with their poison. So previously we said that they were kind of carefully applying it via their mouth. Now they're just spraying huge amounts of poison, venom onto the pupa. And they're also breaking off the limbs of the pupa and other body parts and actually spraying these with acid. So that's what this individual is doing here. It's broken off a leg and it's spraying that leg with acid. And what we did then was to quantify these behaviors before and after cocoon removal. And what we find is that, so that's what this graph shows before and after. And this axis on this graph is the amount of grooming which is occurring. And you can see that grooming increases after cocoon removal. This graph shows poison spraying. So you can see that poison spraying is essentially absent before cocoon removal, but then it really increases afterwards. And the same is true of this kind of biting, this dismemberment behavior. Again, pretty much zero beforehand, but then massively increases once they've removed the cocoon. And what's kind of, this is a very severe response. They're obviously destroying the pupa essentially. But what's more kind of disturbing is that the ants actually perform this whilst the pupa is still alive. So by using a microscope, we could actually quantify how many pupa were alive at the start of when the cocoons have been removed. And you can do that by looking at the dorsal aorta, which is essentially the heart of the pupa. And you can see that pumping here. So it's basically looking at the back of the pupa here at the top of the abdomen. And you can see this dorsal aorta pumping away here. So we use this as an indication of whether or not the pupa were alive or dead. And we found that the majority were alive when the ants started to destroy them. And of course, they were all dead afterwards. And I mentioned that there's a lot of poison spraying going on, and we can actually quantify this as well by measuring the pH of pupa before and after cocoon removal. So this is the pH of pupa before the cocoon's been removed. It's relatively neutral. There's probably been some acid spraying going on the cocoon surface. But once the pupa have been removed from the cocoons and the ants have been allowed to perform this behavior, we see a rapid sort of, or a significant reduction in the pH of the pupa. So they're becoming more acidic. And we can actually visualize what the pupa look like before and after the ants have performed all of these behaviors. So this is 1 hour after the cocoon removal, and this is 24 hours later. And what you can really see is kind of, there's lots of holes, the limbs have been removed, and it's kind of just been chewed up into this kind of pulp. And so we decided the combination of this sort of acid spraying, which we know is a disinfectant, and the dismemberment and wounding of these pupa, we decided to call this behavior destructive disinfection. And we set about essentially trying to understand why the ants perform this behavior. So one of the big questions we had is, how are the ants detecting which pupa are infected? How are they, as I mentioned, the ants are alive when the ants begin to, the pupa are alive when the ants begin to perform this behavior. And there's no obvious signs of infections. They're asymptomatic at this time point. There's no fungal outgrowth or anything. Remember, the fungus has to kill its host before there's any visual signs of infection. And so we suspected that they probably are using some sort of chemical cue. And so to test this, we performed kind of a wash experiment. So this graph shows the proportion of pupa which are being removed from the cocoons when they are unwashed. So that's our control group. This is just the baseline level of unpacking which occurs. And then we either wash pupa in water or a solvent. And the reason why we use a solvent is that many of the chemicals present on the cuticle of the pupa are kind of fatty And so they shouldn't be removed with water, but they should be removed with the solvent. And so what we would expect if there's a chemical cue here is that we see no difference in the amount of unpacking between the unwashed and water groups, but we should see a reduction in unpacking behavior when the pupa have been washed in this chemical solvent and presumably removed the chemical cues which the ants are using to detect an infection. And that's exactly what we see. So this, as I said, this is the key group to focus on here. What we see here is a reduction in cocoon removal when they've been washed in the solvent. And we wanted to go a step further and actually try and identify the compounds which are eliciting this behavior. So we can take these washes and we can use a technique called gas chromatography mass spectrometry, which essentially, you feed it these washes and it identifies all of the different chemical compounds in there. And not only does it identify them, it tells you in what kind of relative concentrations are they present. And what we find is that there's no specific, there's no new chemical present on these sick pupa. So that tells us that it's not simply it's not the pathogen, it's not some odor of the pathogen which the ants are using. It's really coming from the pupa itself. And so what we find essentially is that this is the abundance of these chemical compounds. And in blue, these are the control groups. So these are uninfected pupa. And in green, these are these unpacked infected pupa. And what you can see is that there were 4 compounds out of several hundred, which actually had increased abundances on these infected individuals. And as I said, these weren't novel compounds. They're also present on the healthy individuals, but there's just an increase in them. And we are actually able to show through sort of further experiments that they seem to be linked to the immune response. So when these pupa are infected and they mount an immune response, there's a corresponding increase in these chemicals as well. And more interestingly is that 2 of these chemicals are also found in sick honeybee larvae. And so it's possible that there's some sort of conserved some sort of conserved chemical sickness cue, which multiple social insect species are using to identify sick individuals within their colony. So it really seems then that the ants are able to detect these infections and then they perform all of this destructive disinfection behavior. And next we wanted to know, well, why do they perform destructive disinfection? So to test that, we simply kept pupa which were exposed to the pathogen. We kept them with ants for 24 hours so that they could perform normal sort of sanitary care behavior, and then we isolated them from the ants. So we kept them in separate petri dishes without any ants. And then we did the same with another group where instead now what happened is the ants were allowed to perform also destructive disinfection, and then we removed them from the ants. And in both cases, we kept the pupa, the cocoon pupa and the destructively disinfected pupa separate from the ants in their own petri dishes, and basically just observed how many of those actually then had fungal outgrowth on them. So how many were actually still infectious. And what's really interesting is that as expected, in the group where none of these behaviors were performed, so the pupa which were in the absence of ants, as we said before, about 85–90% become infected, so can have the potential to still cause epidemics. Whereas in the group which have undergone this destructive disinfection, we see a significant reduction in the number becoming infectious. So about 95% no longer become infectious. So through the performance of this behavior, so let me just summarize that because I know it's a lot of information. So what we found then is that the pupa are producing some sort of chemical cue which elicits the cocoon removal behavior by the ants. They then proceed to bite, spray with venom, and dismember these pupa. And this results in essentially sterile, or not necessarily sterile, but no longer infectious corpses, or corpses which no longer can become infectious. So by performing this complicated behavior, the ants are able to seemingly prevent any sort of onward transmission of the disease. But we wanted to understand, so this is a really complicated set of behaviors, and we want to know why do they perform, this complicated set of behaviors. And so we became ants ourselves, essentially. So using sort of micro scissors and forceps, we were able to mimic the cocoon removal behavior ourselves. And then using micro scissors, I also cut up the pupa and sort of mimic the wound in which they perform. And then because we know the chemical composition of the acid, we can recreate that synthetically in the lab as well. And what we see, these are the groups that we had. So we had groups where the pupa was still cocooned, removed from their cocoon or dismantled, and then we treated them with either water as a control or this artificial poison. And this graph is a bit complicated, and so I'll just walk you through it. But essentially what we see is that, so on the bottom here, we've got our cocooned group, our pupa removed from their cocoons, and the removed from their cocoons and destroyed pupa. And what we see, this on the y-axis, this is the proportion of pupa which still become infectious. And what we see is that in all cases, when there's no venom present, the majority still become infectious. But when you introduce the venom, we start to see a reduction in this. And specifically, when we have the combination of all three behaviors, so cocoon removal, poison spraying, venom spraying, and the destruction of the pupa, we see a complete reduction in the number becoming infectious. And so finally then, we wanted to see whether this destructive disinfection actually prevents epidemics within the colony. So we set up these sort of mini nests containing larvae, brood and ants, and we introduced either a sporulating pupa, so mimicking a situation where they might have accidentally missed the pupa and allowed it to become infectious, or one of these pupa which has undergone destructive disinfection. And we quantified then the number of ants within those colonies that actually contracted an infection and became infectious themselves. And so in the sporulating corpse case, what we see is 40% of those workers actually dying from infections and becoming infectious, whereas in the disinfected corpse case, we didn't see any ants contracting the disease. And so just to summarize this all up, this is sort of the normal fungal life cycle of the ants, of this fungus. But what we're seeing is that the ants can perform initially sort of sanitary care to try and this care performing behavior to try and prevent infection. But if that fails, sorry, my slides are being a bit slow. Yeah, if that fails and an internal infection occurs, they can perform this kind of secondary layer of defense called destructive disinfection, which completely prevents any onward transmission. So essentially they're able to stop the fungus's life cycle from continuing. So just to finish then, I wanted to talk a little bit about what we can potentially learn from studying social immunity in ants and what kind of similarities and dissimilarities we see between human societies and ant societies. I think in both cases, we see kind of how preventative measures are really important, as well as good hygiene and the use of disinfectants. In both cases, we also see proactive social distancing playing a really important role. And also things like immunization. So ants and humans seem to rely on, or we know that we rely on immunization to try and control infections or control epidemics. And we also see quarantining as well. But obviously there are some key differences here. So what the ants are really good at compared to humans are detecting infections very early on. We've obviously got better at that during the pandemic with asymptomatic sort of testing with lateral flow tests and what have you, but the ants still, do much better than us there. They're also obligate altruists. So in the same way that it pays a honeybee to, sting a predator and die in the process, it does that for the good of the colony. These pupa which are sick, they signal that they're sick for the good of the colony, because it means that if they're destroyed, the colony still survives. And obviously we wouldn't expect this in humans. So the ants are much more selfless in a way. They also have this kind of, very coordinated, self-organized responses, which don't require any sort of top-down organization or legal rules or anything. It's all completely self-organized without any instructions. And they also have this, care-kill dichotomy. So they will initially care for individuals, but then they also sacrifice individuals when it's no longer possible for them to, to be saved. And obviously we can't do this in human societies. I'm not condoning that we should, just pointing out the differences here. And obviously, yeah, they have these sort of very drastic measures as we've seen. And so perhaps really, maybe the analogy or comparing human societies and ant societies isn't the right comparison to be making. And what I'm really interested in is that potentially these collective disease defenses actually constitute what we might consider actually an immune system at the colony level. And the reason for that is that we can think of social insect colonies as being very similar to a multicellular organism. So in a multicellular organism, such as a mouse or, you or I, we're composed of somatic cells, which don't reproduce. That's things like our skin cells, our organ cells, and then we have germline cells, and they do reproduce. So that's our sort of egg and sperm cells. And that's what comprises a multicellular organism. And in ant societies and other social insect societies, we see the same. So there's a reproductive queen, and then every other individual in that colony is sterile and essentially functions like somatic tissue in our body. So ants kind of act like organs, whereas the queen acts like the germline cells. And in the same way that multicellular organisms have evolved an immune system that prevents them from contracting infections, prevents infections from spreading within the body, and also importantly prevents the germline from becoming infected, we see, or I hope I've convinced you through this talk, that we see very similar sort of layers of defense in social insects as well. And so rather perhaps we can, and there's this really nice quote here from Hamilton, who you probably have heard of before. You've probably heard Hamilton's rule. He's a very famous evolutionary biologist and social insect biologist. In the 80s, he said that, talking about honeybees, he said, nothing like the immune system for detecting and combating microbial enemies is known to exist. There he's referring to, the multicellular immune system. And at the time, people hadn't done any of this social immunity research. And in the decades since, we now know that actually there is something like an immune system in social insect colonies. And perhaps we can talk about a social immune system operating at the colony level. And as we see in our immune systems, when a cell is infected, it actually produces these find me, eat me signals, which elicit that cell's destruction by immune cells. And in ant colonies, what we're seeing is something remarkably similar. So we see that individuals are producing some sort of chemical cue or potentially signal, which elicits their destruction by ants, which kind of act like immune cells. And so subsequently actually, destroy that infection before it can spread within the body. And so that's what I'm really interested in now at Oxford, is actually exploring the idea that potentially we're seeing sort of convergent evolution between multicellular organisms and these kind of superorganism social insect societies, and whether or not they've sort of arrived at sort of similar solutions and immune system to a common problem, infectious disease. And so with that, I'd like to just thank the people which were involved in this work. So the majority of this work I did when I was at IST Austria, where we were funded by an ERC grant, which was awarded to Sylvia Kramer, who was my PhD supervisor. But I've since actually been awarded a John Fell Fund grant to work on this at Oxford. And I'd like to thank St. Hugh's for giving me the opportunity to speak today, and also thank you for your time. And I'm happy to take any questions. So I'm just going to stop sharing my screen now so that I can look at the Q&A box. So there's a number of questions here. So is there an indicator or pheromone that causes the isolation, or is it individual behavior? I assume this is in reference to corpse removal. And yes, there, for example, there's numerous studies showing that when an ant dies, or actually multiple social insect species, when they die, they produce a pheromone which triggers the ants to remove them from the colony. And sometimes it's actually the absence of certain alive signals which also trigger that behavior. So again, similar to what we saw in the ants with infections, they are able to detect who is dead as well. How do you get the QR codes onto the ants? So I should just point out again, I wasn't involved in that study, but you just super glue them on. So you can use cyanoacrylate glue, which is the same glue which surgeons use. So if you might go to the ER room with a cut, and sometimes instead of using stitches, they just use a super glue. And the reason why they use that is because it actually does have I think it has antiseptic properties, but it also is inert, so it doesn't cause any chemical reactions, doesn't release anything into your bloodstream. So we can use that as well with ants, and it doesn't cause any harm to them. Are the people using chemical cues altruistically to flag that they are infected? So I believe that they are, but it's really difficult to discern what is a cue and what is a signal. And there's quite an important distinction between them. So a cue is, any sort of information that an individual inadvertently gives off that others have evolved to recognize. So for example, a cue in humans could be a runny nose, which you involuntarily produce. And that's a warning to others that you might be sick. And so we may have evolved to recognize those symptoms in someone else. Whereas a signal is where you actively are telling, actively giving off information that you are sick. And that's what we might be seeing in the ants, but we can't be sure. So as I sort of said, we've been able to show that this the chemical cues which they produce, or rather have increased levels of when they're sick, they are related to immune response. The way that we did that was to inject them with fungal cell wall components. So we triggered an immune reaction without actually having them be infected. And there was actually no fungal pathogen present. It's just the components of the cell wall of the fungus that we introduced. And that triggered an immune reaction. So we saw increased regulation of immune genes and subsequently caused this increase in their chemicals on their cuticle. So it's possible that there's some pathway there, some connection between the immune response and their chemical odor, which is, potentially could be an evolved signal. And it would actually, as we sort of said, it would pay for these individuals to signal that they're sick, because if they're destroyed, even though they're dead, the colony still survives. So it makes sense that it would be an altruistic signal, but we can't be 100% sure whether or not that's the case. And it's really hard to disentangle those. Do they perform the same destructive process to adult ants or something else? So I didn't mention it, but adult ants, when they're sick, can actually self-isolate. So there's numerous studies showing that when they're sick, they actually remove themselves from the colony, and they'll die, somewhere off in the environment. But that probably depends on the species. So in some of the species where that's been studied, they are very, very small species, very small colonies, and they don't have huge foraging ranges. So there may be less risk that they re-encounter those corpses. But interestingly, in the species we studied in the field, we've actually found graveyards under rocks. And so it really seems like they actually process their dead and are placing them there. So it's possible that they do perform a similar behavior to adult ants, but we don't know actually whether they do. How long does the immunization appear to last? Do the ants ever induce the equivalent of a booster after a certain amount of time? So we don't know. We haven't looked at sort of multiple successive infections, but the work that I was involved in with this was sort of leads on from that. So they have this immunization, and we don't know how long it lasts, but those ants do survive, and they will continue to perform care to others. And so what's interesting is that, and what's really interesting is that they change how they care depending on whether they've got this immunization or not. So when they're immunized, they actually perform much more grooming and sort of intimate interactions with those exposed individuals. But if they encounter an ant which is carrying a pathogen to which they're not immunized against, so a different fungal pathogen, they're actually more likely to die, because presumably, there's some sort of, like, if they get a double infection, it overwhelms their immune system. They don't have any protection against that new pathogen. So if they encounter an ant exposed to a novel pathogen to which they're not immunized, they change their behavior. So they no longer perform so much grooming and like close interaction with the exposed individual, but they actually start spraying their poison essentially from a distance. And so they're still performing care, but they're modulating how they do it to decrease their risk of falling sick. So it's really, really interesting. And yeah, I'd love to know more about sort of how long that lasts and what other kind of impacts it has. So this is the last question. How much does the length of the socialization from the network differ depending on the pathogen? Do there appear to be any pathogen-specific behaviors? We don't know. So majority of this work so far has been carried out with fungal pathogens, but we know ants are infected by viruses and bacteria as well. And you can imagine that their responses would differ depending on the pathogens, because fungal pathogens infect their host by attaching to the surface of the body. So there is a period where they can be mechanistically removed through grooming and chemically disinfected, but viruses tend to spread mouth to mouth and through food can be foodborne, for example. And similarly, many bacteria are also transmitted through food or mouth to mouth. So it would be really interesting to know sort of what's going on with these other pathogens. And I just think there is one study showing that when an individual has been exposed to bacteria, it produces antimicrobial peptides, I believe. So essentially almost like an antibiotic in its saliva or oral liquids that it then can pass mouth to mouth to uninfected individuals. And I can't quite remember now the results of that experiment, but I believe it conferred some sort of protection to those naive sort of non-exposed individuals. So, I mean, it's really amazing. Just to sort of summarize, yeah, I mean, I really think that these social insects, they've had millions of years to evolve and to cope with their diseases. And so it's really not a surprise that we don't see epidemics all the time in their societies. And I do think we have something to learn from them. And I hope that my talk today has been enlightening and enjoyable. And I thank you again very much for listening.
Chris, thank you so much for that. I am, as you know, in no way connected with your scientific fields of interest. I work on European literature, and what I got from that was an enormous amount of scientific insight, but expressed in a way that a complete layperson could understand and follow the argument. The guests here this evening will appreciate that St. Hugh's is very grateful for someone like Chris, because it's obvious he's a great teacher. He does high-level research, he does groundbreaking research, but he's able to present the high-level research at a level that everybody can understand. I would love to be one of his students. I'd love to be in supervisions and tutorials with him to find out more. And we are very grateful, all of us, for your insights this afternoon, this evening. I'm grateful also to everybody who's attended. We had over 100 people present. I'm grateful for everyone who's given donations, and we will continue this series of lectures with the greatest of pleasure. Chris, you set us a very high bar. Thank you.